In this activity, you're going to examine the internal structure of a Eudicot root using images of microscope slides. You've already looked at monocot roots, so now your main goal is to compare how the same tissues are arranged differently in Eudicots. Even though the tissues are similar, the pattern they form in the center of the root is very different. And that pattern is one of the easiest ways to tell monocot and Eudicot roots apart. Eudicot roots share many of the same tissues you saw in monocot roots: an epidermis on the outside, a cortex, an endodermis, a pericycle, and vascular tissues made of xylem and phloem. The big difference isn't what tissues are present, but how they're arranged, especially in the vascular cylinder or stele. That arrangement is what you'll focus on in this activity. In a Eudicot root, the primary xylem sits right in the center and is arranged in a very distinctive cross-shaped or plus-size pattern. Between the arms of that xylem cross, you'll find small groups of primary phloem. The cross-shaped xylem strengthens the root and allows efficient radial transport with phloem between arms. Unlike monocot roots, Eudicot roots typically do not have a large central pith. The vascular tissues occupy the center directly. This cross-shaped xylem pattern is one of your biggest visual clues that you're looking at a Eudicot root. Surrounding the vascular tissues is the pericycle, which sits just inside the endodermis. As you've seen before, the pericycle is where lateral roots originate, and the endodermis acts as a boundary that regulates movement into the vascular system. Outside the endodermis is the cortex, a thick region made mostly of parenchyma cells that takes up a large portion of the root. The epidermis forms the outermost layer and in living roots may produce root hairs to increase surface area for absorption. Before you start analyzing the slide images, think about this question. Why might a cross-shaped arrangement of xylem in Eudicot roots be an advantage compared to the arrangement you saw in monocot roots? Consider things like strength, transport efficiency, and how water might move through the root. Write your prediction in your lab notebook before continuing. We'll start by looking at the cross-section image of a buttercup root. Find the endodermis, which should appear as a band or ring of more darkly stained cells near the center of the root. Everything between the endodermis and the epidermis is the cortex. Typically in Eudicot roots, the cortex is much less than monocot roots. Buttercup is known to have a large cortex. You can see the purple stained amyloplasts inside the cells since they store starch. Now that you're oriented to the outer layers of the root, shift your attention to the center of the cross-section image. Just inside the endodermis, you should see the pericycle. This is a thin layer of cells that surrounds the vascular tissues and is especially important because it's where lateral roots originate. Locate the primary xylem. In a Eudicot root, this tissue is arranged in a very distinctive cross-shaped or plus-sign pattern right in the center of the root. That shape is one of your biggest clues that you're looking at a Eudicot root rather than a monocot root. Next, look for the primary phloem. The phloem appears in small clusters between the arms of the xylem cross. These cells are usually smaller and thinner walled than the xylem cells and may look more lightly stained or more compact. As you're identifying these tissues, use three main visual clues: the shape of the tissue pattern, its position within the root, and the relative size of the cells. These features will help you confidently tell xylem and phloem apart in the slide images. Now, using the slide image as your reference, draw a labeled cross-section of the Eudicot root in your lab notebook. Be sure to include and label the epidermis, cortex, endodermis, pericycle, primary xylem, and primary phloem. Focus on showing their correct positions and overall arrangement, especially the cross-shaped xylem in the center. Let's compare and contrast some tissues you've learned. First, compare the arrangement of the vascular tissues in the two roots. In the monocot root, the vascular tissues form a ring around a central pith. In the Eudicot root, the xylem forms a cross-shaped pattern in the center, with phloem located between the arms of that cross. Think about how these two arrangements are similar and how they are different based on what you observe. Next, focus on the overall organization of tissues in both roots. Using both images, list the tissues from the outside of the root to the inside in this order: epidermis, cortex, endodermis, pericycle, and vascular tissue. Pay attention to whether both root types follow the same general sequence, even though the vascular tissues are arranged differently in the center. Finally, compare a Eudicot root cross-section to a Eudicot stem cross-section. Think about what visual features would help you tell them apart. Focus especially on the arrangement of the vascular tissues and whether there is a central pith or a central xylem pattern. Use those visual clues to explain how you can distinguish a root from a stem and cross-section. To summarize, Eudicot roots have a very distinctive internal structure. The primary xylem forms a cross-shaped pattern in the center, with primary phloem located between the arms of that cross. Unlike monocot roots, Eudicot roots typically do not have a large central pith. Even though the same basic tissues are present in both groups, their arrangement is different, and that difference affects how the root functions and how we identify it. Once again, the big idea holds: structure supports function.